Today's lecture will be about the oncogenicity of viruses, and it will focus on two main themes: how viral infections lead to the development of cancer, and how the research on viruses has led to our better understanding of how cell growth and cell death are regulated. The expected learning outcomes from this lecture include the understanding of the process of oncogenic transformation in the context of viral infection and the different timelines of this transformation depending on the virus, the identification of the transformed phenotype and what do we mean by it, the appreciation of the different mechanisms at play in the transformation by RNA or DNA viruses. You would also become familiar with some viral oncogenes and how they function. As an example, we look at a human T-cell lymphotropic virus, HTLV, as a model for RNA tumor viruses that can lead to the development of leukemia. We will also review the human papillomavirus, HPV, a DNA tumor virus, and how it can lead to the development of cervical and other cancers. Finally, you will recognize the importance of prevention of HPV infection by vaccination and its remarkable success in countries that have implemented routine vaccination on their schedules. So during homeostasis, the cell machinery that is necessary for replication of cells, for processing RNA and DNA and making new protein, is constantly regulated by signal transduction pathways that start from extracellular or intracellular growth signals that might involve receptors, and then might involve signal transduction pathways that regulate the cellular machinery. Occasionally, cells may receive death signals, that is, signals that might order the cell to commit suicide, to undergo apoptosis, and these could also be either internal or external. Now, viruses, on the other hand, they prefer to have a machinery that is subverted, that is, it's working under their own terms, under their own orders. They want the machinery to be busy making new virus, new viral proteins, new viral RNA, or new viral DNA, depending on the viral types. So for that purpose, viruses will make different kinds of proteins that help subvert the different signal transduction pathways, whether they are growth pathways or death pathways, and sometimes both. The purpose remains the same, which is to make more virus. Now, the concept of oncogenesis is basically the transformation of a normal cell into a cancer cell. And in most cases, this is a multi-step process. It doesn't happen with a switch or a one-step process. In the majority of cases, it's a multi-step process. And the infection with a tumor virus, in general, represents only one of the steps in the oncogenic transformation. In general, there is a long latency, that is a period of months or years, between infection and tumor development. Another concept that is of importance is that cells can have either a permissive or a non-permissive infection. The permissive infection, also known as a lytic infection, is when the cell is an excellent host for the virus, and the virus produces so much progeny, so much viral particles, that eventually the cell lyses, that is, it dies, it bursts open. So that cell is lost and cannot be transformed further. whereas in the non-permissive or non-lytic infection, the cell may continue to survive, but the presence of the virus will manipulate it, will change it in certain ways that over time it might transform into a tumor cell. Now here, the immune system plays an important role. The immune system usually gets rid of cells that are infected with either viruses or bacteria or fungi. This is part of its job. And when the immune system detects a cell that is infected, whether permissively or non-permissively, it tries to get rid of that cell. And this is very relevant for cases with immune deficiency. For example, patients who are born with a dysfunctional immune system are at much higher rates of viral induced cancers during their lifetime. Now, once a cell is transformed, there are several features that can be attributed to that transformed phenotype. First, there is an increase in saturation density. That is, the number of cells per square centimeter increases. Normally, cells, when they grow, they reach a saturation point where they touch each other and they can no longer proliferate. That is what saturation density represents. So in the transformed type, there is an increase, that is a doubling or tripling of the number of cells that can be present in a certain area. They can also have a reduced growth factor requirement, which means that they are somewhat independent of growth factors. They are on automatic growth mode. They don't require the growth factors to push them to grow. Also, they require a lot of nutrients because they are replicating faster and making more daughter cells. They are in need of nutrition, of glucose, amino acids, and other nutritional support. They are also anchorage independent, and this is a feature of tumor cells that can grow in a liquid medium where they can replicate even if they are not anchored to a solid surface. And they produce a fair number of proteases that help them detach from their solid surfaces that they are adhered to. So this helps them not only replicate better, but also to spread in the body. So tumor viruses are those viruses that can lead to the development of cancers. And in general, these viruses can cause a persistent long-term infection in the human host. And in general, they have oncoproteins in them that interact with the cellular proteins and cellular signaling pathways to make viral reproduction easier and more efficient. And as a side effect, they end up causing cancers in some situations. And these are several examples of such tumor viruses. The papillomavirus, the human papillomavirus, can cause cervical and anogenital cancers. The human T-cell lymphotropic virus type 1, which is related to the HIV virus, which originally was called HTLV3, can cause adult T-cell leukemia or lymphoma. Hepatitis B virus can cause liver cancer. Hepatitis C virus can also cause liver cancer and B-cell lymphoma. Epstein-Barr virus can cause Hodgkin lymphoma, non-Hodgkin lymphoma, and nasopharyngeal carcinoma. And the human herpes virus 8 can cause Kaposi sarcoma and primary effusion lymphoma. So first we'll talk about the RNA tumor viruses. All of the RNA tumor viruses are retroviruses and they cause non-lytic infection. The majority cause tumors in birds or mammals other than humans. The only human example is HTLV1, which we'll discuss later. And the tumor latency in these viruses is usually days to years. So RNA tumor viruses may have oncogenes, and these are not essential for viral replication, and in fact are initially cellular genes that were acquired by the retrovirus when it integrated with the host genome and then got picked up and became part of the viral genome. Frequently, actually, their presence is detrimental to the viral replication. So, in their presence, some of these viruses may depend on helper viruses that are lacking in the oncogene in order to replicate efficiently. So cellular oncogenes are conserved over long evolutionary distances, and they play an essential role in cell regulation. Their carcinogenic potential is actually due to their dysregulation, to their corruption of the physiologic function. The oncoproteins that are encoded by these oncogenes are components of signaling networks that help convert extracellular signals into patterns of gene activity. So when you compare the cellular oncogenes with their viral counterparts, you find that the cellular oncogenes play a physiologic function in regulating the cell, whereas the viral counterparts are usually truncated or mutated, and this leads to their dysregulation. The dysregulation means that the viral oncogenes are functionally constitutively active, so they are always on, and this can lead to transformation. So there are several mechanisms by which RNA viruses produce oncogenicity. First, there are the transducing retroviruses, which carry an oncogene within their genome. So retroviral transduction is actually an infrequent form of transformation where it is exceedingly fast. It happens over a few hours or days, and it results in polyclonal tumors because it's a single-step form of carcinogenesis. The mere expression of the oncogene, the viral oncogene, leads to transformation. So this, as I mentioned earlier, is the less common form of transformation compared to the multi-step process that normally occurs in humans. The transduced oncogenes are basically necessary and sufficient for transformation. This slide is important only in illustrating the variety of viral oncogenes that we have encountered. And this is important from two aspects. One, to know that the viral oncogenes can be growth factors, such as the platelet-derived growth factor, or there can be receptors or enzymes, such as the GTPase or the tyrosine kinase, or the serine threonine kinase, or they can be transcription factors, such as AP1, MYC, and NF-kappa B. Importantly, all of these oncogenes were crucial when they were discovered in the understanding of their cellular counterparts, the cellular oncogenes or proto-oncogenes that were in many instances unknown until the viral oncogenes were discovered. Of course, you don't need to remember any of these specifically, but just to learn what the variety is about. Now, another mechanism of oncogenesis is the cis-activating retroviruses. These viruses do not carry an oncogene, but they may integrate in the vicinity of a cellular oncogene, and as they are replicating, they will inadvertently activate the cellular oncogene and lead to transformation. The third mechanism of oncogenicity is by transactivating viral proteins. So these are non-structural viral proteins that are transcriptional regulators of one or more cellular genes that are involved in growth regulation. So when they are expressed, they drive these cellular genes to help the cell grow and lead to transformation. So the first example we'll discuss today is the human T-cell lymphotropic virus type 1 and its relationship with adult T-cell leukemia or ATL. In all cases of ATL, we can find provirus of HTLV1, that is incomplete virus. There is no active viral expression in ATL. This is a monoclonal disease that occurs usually with a latency of about 20 years and occurs only in about less than 1% of infected patients. So there are multiple steps that are required in the transformation of infected cell to a tumor cell. There is no transduced oncogene in the HTLV1 genome, and there is no preferred integration site. So it's not through cis activation. This is an example of transactivation that results in the transformation of the host cell. The viral protein that plays an important role in the replication and in the transformation of HTLV1 infected cells is the TAX protein. This is a non-structural but essential protein. It has no cellular homologue, so it's a unique kind of a protein, and it's a transcriptional activator of the HTLV1 as well as multiple cellular transcriptional factors, including CREB, ATF, and NF-kappa B, which are very important for the transcription of a number of growth factors and cytokines in the cell. So this is the structure of HTLV1. It's very similar to HIV, but has different surface and matrix proteins and lacks an integrase. It's a relatively rare infection in the world. About 15 to 20 million people are infected worldwide. There are regions of high prevalence, such as south of Japan, the Caribbean, and tropical Africa. Transmission is by sexual contact, breastfeeding, blood transfusion, and sharing of needles during drug abuse. So the two clinical presentations that are associated with this virus include the T-cell leukemia lymphoma, which we mentioned occurs in less than 1%, and tropical spastic paraparesis, which is also a rare neurologic presentation. Most of the infections are actually asymptomatic, and they occur after sexual contact or blood transfusion from an infected individual. And the virus infects CD4 positive T cells, and as with retroviruses, the provirus integrates into the genome and viral genes become expressed, including the TAX protein, and there is a subsequent production of cytokines, cytokine receptors. IL-2 promotes the proliferation of these cells, the CD4 positive T cells, and they start rapidly dividing, and over time they accumulate mutations, and that may lead to transformation into a T-cell leukemia / lymphoma. The viral oncoprotein TAX has been shown to have a major role in cellular transformation. The diagnosis of HTLV1 infection is usually by demonstrating antibody titers in serum or CSF, or of course by PCR. Treatment of the leukemia is by chemotherapy. There is no definitive regimen that has been demonstrated to increase the median survival time. This is a difficult to treat tumor. Relapse is common. There's been some promise using antiretroviral treatment that has been shown to be as good, if not better, than chemotherapy for the leukemic form. Also, retinoids or arsenic have been shown to be promising new modalities in the treatment of HTLV-1. Steroids for the myelopathy are among the potential treatments. Next, we'll move on to discuss DNA tumor viruses. We mentioned before that there are several DNA viruses that are associated with human cancers, including EBV and Burkitt's lymphoma, HPV and cervical cancer, hepatitis B virus and hepatocellular carcinoma, and HHV8 and Kaposi sarcoma. There are other DNA viruses that cause cancers in animals, but not in humans. Adenoviruses and polyomaviruses can cause various solid tumors in animals. Pox viruses can cause myxomas and fibromas. Now, DNA tumor viruses usually have oncogenes. And unlike the RNA tumor virus oncogenes, DNA tumor virus oncogenes are essential for viral replication, and they don't have any cellular counterparts. So they are unique proteins that are only found in these DNA viruses. Now, infection in permissive cells usually results in a productive infection followed by cell lysis, so that transforming events usually are missed and you don't get a cancer as a result. Whereas the infection in non-permissive circumstances is more likely to be followed by transformation. Now, the targets of DNA viral oncogenes include the regulators of the cell cycle machinery, such as the retinoblastoma protein and P53, apoptosis machinery, such as caspases, which are proteases that are important in the signaling and execution of apoptosis, and the BCL-T family of proteins, which is a large family of proteins that can either inhibit or promote apoptosis. Now, first, to discuss the retinoblastoma protein, RB. This is a large nuclear phosphoprotein that plays an important role in growth suppression. So it's a tumor suppressor protein. And when both copies of RB are lost, either genetically or by mutation, that can result in cancer. And this protein is regulated by phosphorylation. So the hypophosphorylated form correlates with keeping the cell in the G0, G1 phase, that is arrested in this phase of the cell cycle, or with the differentiated phenotype. So RB is very important in regulating the cell cycle. And the reason it's important is that it regulates an important transcription factor called E2F. And the way RB functions is that when it's active, it binds to the transcription factor E2F and prevents it from promoting gene transcription. When RB becomes phosphorylated, it releases E2F, and E2F can then become free to promote gene transcription and cell cycle progression. P53 is another tumor suppressor that functionally is altered in many human cancers. It induces cell cycle arrest by activating RB, and it can induce apoptosis in response to severe injury, such as when there is an infection, viruses or otherwise. So in the context of viral infection, the virus prefers to see a cell that is replicating. That is a much better environment for viral replication. So a virus is not happy to see RB active. So several viruses actually make proteins to inactivate RB so that RB would release E2F so that E2F can be used for gene transcription, both cellular and viral transcription. And here it's important to note that the name of this transcription factor, E2F, was actually coined during research on adenoviruses, where it was coined the early adenovirus protein 2F. And this turned out later to be actually a cellular transcription factor and not a uniquely viral transcription factor. And not only adenovirus targets RB, but there are other viruses that also make proteins that target RB, including the large T protein of polyomaviruses, the E7 protein of human papillomavirus, and the EBNA5 protein of Epstein-Barr virus. Now, if you look at specific viruses, you'll find that a certain virus may have more than one oncogene that can help it promote transformation. The example of Epstein-Barr virus, it makes several proteins. EBNA5 inhibits RB and P53. EBNA4 simulates BCL2. BHRF1 simulates BCL2. And the LMP1 activates BCL2 and NF-kappa B. All of these help the virus promote cell proliferation and inhibit apoptosis so that the cell survives long enough to make more virus. The adenovirus also makes several proteins. We already discussed E1A that inhibits RB, but also adenovirus makes E1B55K, which inhibits P53. So here it's inhibiting two proteins that are involved in the cell cycle. Also, it makes a protein E1B, which is a similar protein to BCL2, so that inhibits apoptosis, but also it inhibits NF-kappa B. Another two proteins, E3 14.7 and 10.4/14.5, both of these can inhibit apoptosis. So you'll see the variety of proteins that can be made by a single virus to inhibit apoptosis. Now, we talked a little bit about P53, but let's review what P53 does in regulating cell cycle arrest and apoptosis. Now, the cyclin-dependent kinases are enzymes that phosphorylate RB, and when they phosphorylate RB, RB becomes inactive and E2F transcription factor is released to promote cell cycle transcription progression by gene transcription. Now, when there is stress, P53 is upregulated, and if the stress is minor enough, the P53 will induce the inhibitor P21, which inhibits cyclin-dependent kinases. And when these kinases are inhibited, RB is no longer phosphorylated and becomes active so that it can bind to E2F and prevent cell cycle progression. This defense mechanism by the cell is meant to allow sufficient time so that the cell arrests in G0, G1 and repair whatever damage has been encountered on the cell. Now, if the damage is severe enough, then P53 might induce apoptosis by alternative pathways that are independent of RB. So in the context of viral infection, the virus, of course, would not be happy to see P53 drive the cell to apoptosis, because then the virus cannot use the cellular machinery to replicate. So viruses have evolved proteins that block the function of p53. We mentioned the adenoviral protein E1b55K, which can bind to p53 and inactivate it. Similarly, E6 protein of human papillomavirus and EBNA5 of Epstein-Barr virus can both bind to p53 and disable it as well. Now, when p53 is disabled, then it cannot arrest the cell in G0, G1, so the cell now is able to proliferate even in the presence of a viral infection, a very stressful event, and the cell cannot undergo apoptosis. So by doing this, the virus can now block both apoptosis and cell cycle arrest so that the environment is conducive for viral gene. So after discussing the basics of DNA virus transformation, we're going to talk about real-life situations involving DNA viruses and transformation. First, a real-life example, case 1, Ragad, a 25-year-old woman, recently married. She presents to the gynecologist with itching around the labia for several weeks, and several small lesions are seen. One sexual partner, her husband. Case 2, Jad is a 23-year-old man, sexually active, rarely uses condoms. He is presenting with several small lesions on his penis for 2 weeks. Both of these people have warts. These are HPV-induced warts. Warts are tumors, benign tumors that grow on the skin, in this case, the genitalia. And these are some examples of human papillomavirus-induced warts. They can be acute, and they occur usually on the genitalia when they are sexually transmitted, or they can occur on fingers, hands, soles, and knees, and other parts of the body when they are not sexually transmitted, such as in the case of the common warts that are seen in children. This is an example of condyloma acuminata. They can be quite severe, quite disfiguring, and very annoying and difficult to treat. So let's review what the papillomavirus is. It's a DNA virus, of course. It's naked. It has an external capsid, an icosahedral capsid. And it replicates in the nucleus of the host cell. Now, there are several types of HPV. Some are low-risk types that cause benign warts. Some are intermediate to high risk that may result in malignant neoplasms, such as cervical cancer, head and neck cancer, and oropharyngeal cancers. Now, for HPV, humans are the only known reservoir. Now, there are other papillomaviruses that infect species that are other than human, but these are not our concern now. But if you look at the phylogenetic tree, you find that the oncogenic types are, in general, related to each other. You see them on the slide as the gray circles. And in those two circles, the most important types, 16 and 18, are the most common causes of cervical cancer. The other low-risk types are the ones that cause warts, either genital warts or warts on the hands and feet. And among those, the 6 and 11 types are the most important causes of anogenital warts. Now, if you look at the pyramid of HPV-related diagnoses, and this is data that reflects what happens all over the world, you find that there are incidental 660 million HPV infection every year. There are about 30 million infections that result in genital warts, and 90% of those are caused by types 6 and 11. And there are about 30 million infections that cause low-grade cervical lesions, and 30% of those are attributable to HPV types 6, 16, and 18. And then there are about 10 million cases of high-grade precancerous lesions that require treatment, and about 50% of those are caused by HPV types 16 and 18. And then there are about 500,000 cases every year of cervical cancer, and 70% of these are due to types 16 and 18. When you look at genital warts in general, this is a disease of young people. The highest incidence among females is in the 16 to 19 age bracket, but it's also common in young adults, both males and females. So this figure is from a US high school where they interviewed students about their romantic or sexual relationships with other students, and they came up with this diagram, which illustrates that if one of these students has had HPV, genital HPV, it would have been quite easy to transmit to multiple other students. Now, if you look at the human papillomavirus structure, you'll find that it has this external capsid, which is composed of two proteins that are late expressed or structural proteins L1 and L2. And for the different types of HPV, you'll have different types of L1 and L2. And the DNA, of course, is circular and is encapsulated inside this capsid structure. So this has been the basis of the development of the vaccine. The virus is encountered, for example, in this case, in the cervix through a sexual relationship. During sexual intercourse, there might be breaks in the cervical epithelium, which would allow the virus to enter to the basal layer of cells and start an infection there. As the infection progresses, the top layers start to get infected, and as they get infected, the viral replication is completed and virus is shed out of the top epithelial layer. And this leads to a spread of the infection to other parts of the cervix. And of course, if there is additional intercourse with a different partner, that partner may also then acquire the virus. Now, to the left of the slide, you see the relevant proteins from the virus that are important for this viral cycle. And you notice the two proteins we mentioned before, E6 and E7, which are respectively able to bind to P53 and RB and to inhibit them so that the cell can progress and replicate and make more hospitable environment for the virus. Now, these proteins, E6 and E7, are responsible for the transforming and immortalization of cells, which may later lead to the development of cervical cancer because they are able to interact with RB and P53. Now, if you look at the transformation and progression to cervical cancer, you'll find that this takes years to occur. So, if you look at this slide, when the virus comes in to a normal epithelium, this is what normal epithelium would look like in the cervix. Over time, transformation might occur. So you'll see coilocytosis, which is the enlargement of the cytoplasm and the perinuclear space. And this is what you will see on colposcopy. CIN stands for cervical intraepithelial neoplasia and is graded as 1, 2, and 3. 1 is low grade, 2 and 3 are high grade. And this is what the cervix would start looking like. And this process might take years, but as long as you are in CIN 1, 2, or 3, this can be treated usually locally, and the process can be reserved. But once you get to invasive carcinoma, then you have to treat this as invasive cancer with radical surgery and chemotherapy. The important thing to keep in mind is that this is a multi-step process that requires other genetic hits to complete transformation. Now, if you look at the different papillomavirus types, you'll find some of them cause benign tumors, which are the regular warts, the common skin warts. Some are kind of a gray zone, which are usually mostly benign but can progress to malignancy, and you'll find these in the middle of the slide. And then at the bottom of the slide, you'll find the types that can cause high-grade dysplasia and carcinomas of the genitalia and the larynx and the esophagus. And these are, of course, just to indicate the different types of HPV and not for you to memorize. So for the diagnosis of papillomavirus infection, it really depends on the location. For those lesions that are skin warts, usually a simple examination is sufficient to make a diagnosis. But for those lesions that are on the cervix or in the rectum, usually we have to apply 1% acetic acid, which will turn the infected lesions white, and those can be then biopsied so that their pathology can be examined in detail. So this, when used in the cervix, is called the Papa-Nicolau testing or the Pap smear, which has been effective over the years in detecting dysplastic HPV infected lesions so that they can be treated or removed before they develop into cancer. Unfortunately, not enough women go regularly to the gynecologist to have this Pap smear done, and still a large number of cervical cancers occur every year. PCR, of course, can be used to detect viral DNA and even to determine the specific type of HPV that has caused the infection. Now, for treatment, we have to keep in mind that warts in general can resolve spontaneously within 1 to 2 years. That is true for most skin warts and even for the majority of genital warts. But because these are disfiguring or they may be painful in some situations, they can also be treated. Treatment can be by applying chemical agents such as podophyllin, trichloroacetic acid, or 5-fluorouracil epinephrine. These in general cause irritation, and the irritation recruits immune cells, and the immune cells then can detect the HPV that's been hiding inside those infected cells, and once they are detected, they can be destroyed and eliminated. Immune modulatory agents such as imiquimod or interferon alpha injections can also be used, particularly for the larger lesions such as condyloma acuminata. Surgery usually is reserved for the larger lesions, and for that, cryotherapy or using liquid nitrogen guns or surgical excision or laser guns can be used to remove the large lesions. Now, because screening and surgery were not that effective in controlling the incidence of cervical cancer and other HPV-induced cancers, a vaccine was developed. Now, vaccine development was actually a breakthrough because it was a unique and new technique in development of vaccines. So, in short, what was accomplished is that the outer capsid of the HPV virus was reconstituted in the lab using the L1 and L2 proteins, which make up the capsid protein, so that the manufacturers could synthesize an empty capsid, basically a shell of the virus that lacks any DNA. So the shell of the virus, or the empty non-infectious virus-like particle, it looks like the virus, so the immune system would recognize it and would think that it is a virus, would make antibodies against it, and this shell is empty, so it doesn't cause any infection, it doesn't cause replication or any untoward. So vaccination was supposed to prevent infection by preventing the virus from entering the cell. Now, this is based on experiments that were done in animal models, where it was shown that if the animal had sufficient serum neutralizing antibodies that were circulating, these were enough to prevent infection by HPV virus if it was encountered. So, if you induce sufficient neutralizing antibodies by a vaccine, those antibodies would prevent the HPV virus when it is encountered, would prevent it from causing an infection. And if you give the animal antibodies against HPV, that animal would be protected against HPV infection. So in order to prevent HPV infection, for sexually acquired HPV, of course, sexual abstention would be best. But it's important to also keep in mind that safe sex, as in wearing condoms, does not fully protect against HPV. Both the male and the female partners can still acquire HPV despite wearing condoms. Now, vaccination is another way to prevent HPV infections, particularly the sexually acquired HPV infections. There are currently two U.S. Food and Drug Administration approved HPV vaccines. These are the Gardasil 9, which helps prevent infection with 9 HPV types that include 6 and 11, which are the ones that cause most of the genital warts. And then the remaining 16, 18, and the rest are oncogenic types that are responsible for HPV-induced cancers, cervical and other genital and oropharyngeal and esophageal cancers. Cervarix is the other vaccine that contains only two types, types 16 and 18, the oncogenic types. It's a very effective vaccine, but has the more limited coverage. Now, the recommendation is to give the vaccine for very young people. So it's approved from the age of 9 years and up, usually for both boys and girls to get it, so that they will be protected against both the oncogenic and the wart-causing types. Most schedules prefer to give the vaccine before the age of 15. If it is administered before the age of 15, the vaccine can be given as a 2-dose schedule, about 6 to 12 months apart. If it is administered after the age of 15, then a 3-dose schedule should be given. Although there is recent data to suggest that even after a single dose, there is a lot of protection that can be sustained for a fairly long period of time. But still, as of today, the recommendation is to give 3 doses after the age of 15. In Lebanon, the academic societies that are concerned with the vaccination against HPV have recommended vaccination of all girls at 10 to 12 years of age, although in our private practices we also offer the vaccine to boys as well, to protect them against what we know later on might be a very disturbing encounter with either genital warts or genital cancers of all sorts. And I have to say here that the HPV vaccines have been very effective and very safe in controlling HPV related disease. And of course, they will be more effective if given before any sexual activity. So the younger the age, the better it is to start vaccination. And this will be the end of this lecture. I know it contained a lot of information, but you don't really need to memorize or to learn all of the details about the different viruses, the different proteins, etc. All I want you to remember is really the concepts behind transformation, the different mechanisms by which viruses can cause transformation, and to get a flavor of the clinical impact of these tumor causing viruses. And thank you. So in this presentation, we reviewed the process of oncogenic transformation in the context of viral infection, mostly a step-by-step process that may take several hits to achieve transformation, but also a single-step process, as in the case of some avian RNA viruses. We also reviewed the characteristics of the transformed cell phenotype and how transformation mechanisms differ between RNA and DNA viruses. We also reviewed a long list of viral oncogenes as well as some cellular proto-oncogenes. Here, I should ask you not to memorize each of these oncogenes and how they work, but just to get familiar with the scope of their functions and be at least familiar with their names. We also reviewed how HTLV may lead in some cases to the development of adult T-cell leukemia, and how HPV may lead to the development of cervical and other cancers. We went over the development of the HPV vaccines, a remarkable scientific breakthrough at the time, and the dramatic results seen in countries that introduced HPV vaccination on their schedules, where they have witnessed the near elimination of new cervical cancer cases caused by vaccine types.